Okay, good afternoon, everyone. Welcome to this week's Joint HPL and PCSA Seminar. It's my pleasure today to introduce Dr. Tim Butterfeild. Tim completed his first undergraduate degree in biology and chemistry in Potsdam State University, located in upstate New York. His second undergraduate degree was in sports medicine at Messiah College in Pennsylvania. Between 1990 and 1992, Tim completed a master's degree in athletic training at Old Dominion University. This is located in Virginia. Tim joined the HPL as a doctoral student in 2001, and he studied muscle mechanics with Dr. Walter Herzog. Here he was given the Perrin Award for his outstanding doctoral dissertation by the National Athletic Trainers Association in 2006. His postdoctoral work was done between 2005 and 2007 at The Ohio State University. Here he studied eccentric exercise-induced inflammation in skeletal muscle. Here he was given a research endowment by the American College of Sports Medicine in support of his work. In 2007, he gained a position as a professor at the University of Kentucky in the Departments of Rehabilitation Sciences and Physiology. The lab he established integrates muscle physiology and muscle mechanics in order to study exercise-induced adaptations and methods used to manipulate these adaptations in order to improve muscle function. Tim's published 30 articles and over 100 conference abstracts. In 2016, Tim was awarded the Richard E. Kingston Award for Excellence in Teaching by the UK College of Health Sciences and the Marjorie A. King Research to Reality Award by the Eastern Athletic Trainers Association. In 2013, Tim was awarded the New Investigator Award by the National Athletic Trainers Association. Some highlights of his research career thus far include improving the design of baby bottle nipples to match tongue forces in preterm infants, investigating how massage influences muscle damage and injury, the creation of a inducible titin kinase knockout mouse, and the investigation of larynx muscle plasticity, and that's going to be the subject of today's talk. Now, although Tim no longer lives in Calgary, he retains his love of mountains, but can't spend much time in mountains in Kentucky. So instead, he spends his time woodworking, and he enjoys the challenge of raising 2 German Shepherd and 2 human children.
Thank you, Ian. It's awesome. Yes, and it's a pleasure to be here. Thanks for coming, and thank Maria for a wonderful defense this morning and being the reason that I get to be invited up here in the first place. So thank you all so much. Well done. So my talk today is on laryngeal muscles, and I start out with skeletal muscle, of course, and I wanted to present something that we're currently in the middle of. And so I preface this by saying I'm not a speech pathologist, but I work with many. So please, I welcome any input. These data are incomplete, but they're data, and they're not all even complete, and they won't be all presented today. But I'll present what I have so far. And I have a history of doing titles like this, and if it confuses anybody, I apologize. And it did confuse somebody last night, so I have to explain that this is Pavarotti, the original Pavarotti, and what we're doing is making a Pavarotti. Right? So Mickey Mouse, for those that are too young to know who Mickey Mouse was. The laryngeal muscle group at University of Kentucky was started by Dr. Joe Stemple, and he is the speech language pathologist. He is a member of the American Society for Hearing, Language, Phonation, etc. He is the chief clinician at the Speech and Language and Hearing Clinic, and along with the other 4 people up there, we have been tasked with trying to find a better understanding of the physiology of the laryngeal muscles. Joe likes to say this all the time, every time we have a presentation, which is laryngeal muscles are just like any other skeletal muscle in the body, and the fact that they can be weakened, they become strained, they become injured, and you can remedy that situation by rehabilitation. And his big argument always is there's no evidence for that. So laryngeal muscles are really understudied. There really hasn't been a whole lot of research on laryngeal muscles. We have started doing more research in the last 10 years, but before that, there isn't a whole lot known about them. They are 13 muscles, 6 paired muscles, and 1 muscle that acts by its lonesome. They're very important for not only respiration. The intrinsic muscles are involved in speech. The extrinsic muscles will raise and lower your cartilages, but the internal intrinsic muscles are responsible for phonation, the respiration, and also I'm missing a word. It'll come to me a little bit. Sorry. Here's 5 muscles that are of most importance that we look at. So the cricoarytenoid muscle, the cricoid cartilage and the thyroid cartilage make up the cartilages of your intrinsic laryngeal muscles. And the cricoid cartilage being round cartilage, the thyroid cartilage sits around it and actually articulates with it, and together they'll move like so. And it's the cricothyroid muscle itself, aptly named, attaches to both, that's going to raise and lower your voice box. The muscles on the more interior side include muscles that will abduct your vocal folds. So these 2 lines here will represent your 2 vocal folds. The arytenoid cartilage kind of sits on top of your cricoid cartilage and will rotate. And so the posterior cricoarytenoid muscle, the arytenoid cartilage, when it contracts, it will rotate and it will actually abduct your vocal folds. So it's most important for breathing because it holds that open. The lateral cricoarytenoid and the transverse arytenoids will adduct the vocal folds. And the one that we look at most, because it's the largest of the bunch, is the thyroarytenoid. That one tends to change the tension of your vocal folds, to change the resonance frequencies during vibration. So here's 2 images for you. On the left is a normal closure of a human vocal fold. So there's a stroboscope that will go down posterior of your uvula, and you can see that there's good closure of this one. This is what's called presbylaryngeus or presbylarynx, and that occurs when the muscles, for some reason, are not facilitating direct closure of your vocal folds. And 65% of all people that go into the speech clinic, 65 years or older, will have presbyphonia and presbylaryngeus. The idea is, though, that is due to muscle weakness. The challenge is it's never been shown. So all the therapy that's done for this condition has no evidence to back it. It does work, though. The outcomes of speech therapy are pretty good, but nobody really knows why. So you have all these patients doing all these exercises, and they have pretty good outcomes. But the idea is if you can modify the exercises to optimize the outcomes, that would be a better way to go. So this is a video of the clinic. You'll hear somebody maintaining a good pitch. It's not me, because it wouldn't be me. It's actually Joe, that's what he does. And you'll hear the pitch that Joe puts it, that gives the patient, and you'll see the patient trying to match that pitch, and you'll see the vocal folds acting to do that. You'll see them closing and vibrating.
And breathe.
And this is what speech therapy entails in the clinic. But you'll also notice that those vocal folds are failing to actually meet, and that limits the frequency that this person can actually hit. So, clinical practice for voice disorders. Again, it's always been assumed that these muscles have the same physiology and biology as skeletal muscles. And again, it's a huge assumption. There's also an assumption that if you do exercise them, that they will increase in mass, that they'll increase in force, that you can change the endurance of these muscles through exercise. And again, even though the clinical outcomes are decent, the physiology and the mechanics have never been shown. Most of these treatment approaches that are applied today then are not based on any of this sound scientific evidence, right? So this is the central question that drives the Laryngeal Physiology Research Group at the University of Kentucky is the biology of laryngeal muscles the same as that of skeletal muscle? And if not, how is it different? And can we optimize that physiology in some way? So this is an image from a development paper about 15 years ago, just to show you that the lineage of laryngeal muscles is completely different than that of skeletal muscle. Skeletal muscle comes from the trunk mesoderm, from the somites, and it is completely separate from the cranial mesoderm, which is where the actual intrinsic muscles of your laryngeal muscles come from. It's also been shown that they don't not fully innervated even until after birth, and some people now propose that some of the modulation and use of these muscles after birth can also affect how they actually finish forming inside of the voice box to improve function. Because they come from a completely different area during growth, they have completely different functions and proteins. So for instance, we did a study about 10 years ago, 8 years ago now, that looked at muscular dystrophy rats and mice. The observation became that people with muscular dystrophy could still speak very well. But as their limb muscles start to fail and they have this necrosis and this dystrophy that occurs, they can still talk. So looking at the actual MDX mouse, we looked at the gastrocnemius of MDX mouse and a control gastrocnemius, as well as the thyroarytenoid of the laryngeal muscles of the MDX and the control, and one cricoarytenoid. And what we found was the laryngeal muscles are spared during muscular dystrophy for whatever reason. And what you find if you look further is because the anchoring protein to the sarcolemma of skeletal muscle is dystrophin, but in laryngeal muscles it's utrophin. So the dystrophin protein loss in dystrophy does not affect your laryngeal muscles. Also, there's a really nice paper that came out in Feng group back in 2012. This is a laryngeal muscle fiber. This is one of the thyroarytenoid fibers from a mouse. And this is immunohistochemistry, so you'll notice the fiber is a muscle fiber. You'll notice the blue are DAPI, those are nuclei. And immunohistochemistry with alpha-bungarotoxin, FTSE conjugated, to show that you have neuromuscular junctions. So these fibers of laryngeal muscles have more than one neuromuscular junction. They also have more than two. This one particular one had more than three. But the image here, of course, only has three on there. So they have multiple innervations by alpha motor neurons, whereas skeletal muscle has only one. And why that is, at this point, nobody really knows. So we wanted to test these laryngeal muscles the same way that we test skeletal muscle. We wanted to subject them to the same sort of exercise paradigms and programs that we've done in the past with skeletal muscle, and we chose rats to do this with. In part because we already had antibodies set for rat use, and we've already done some rat work in the past. And so when the Laryngeal Physiology Research Group came to me and asked if I would help them out, and I said I'd be happy to give this a shot because it's really cool and fun, and why wouldn't you? And so we took some rats, we used 6-month-old and 30-month-old Fisher Norway Brown 344 rats from the NIA, so they're aging model of rat. These rats, when they turn 36 years old, are essentially over 90 in human years. So when they reach 30, they're aging. They're well over 65 years old at the age of 30 months. And 6-month-old rats are about early college age rats. So we have young, we have old. And we had two conditions. We have some control groups, and we have some exercise groups. And each group was broken up into histology and function. We couldn't do both. So we doubled our rats and used histology for half the group and function for the other half. And so we had exercise groups and controls. And for this particular study, the right thyroarytenoid, I said tibialis anterior again, because I did tibialis anterior my entire life. So this is just a new TA. Always do TA. So the right thyroarytenoid muscle is the one that we're using for these studies. We instrumented that side as opposed to the left. So left side will be the contralateral control. We also have pure control that we didn't do anything with except instrument them. So in a cross section of your laryngeal muscles, this is anterior and posterior, your thyroid cartilage and your cricoid cartilage. And the thyroarytenoid runs from your thyroid cartilage to that arytenoid process, articular cartilage. And it's made up of 3 different sections. It has a very long lateral section, very long middle section, and a very oblique pinnated portion right in the middle, right by the arytenoid cartilage, that we kind of ignore when it comes time to talk about the numbers because it just makes things more difficult. But there are 3 sections, 2 very large, 1 very small one. And so we've always made nerve cuff electrodes. I learned them from Tim Leonard and Walter Herzog when I was here. And the ones we made when I was here were these rat peroneal nerve cuffs. We put them on sciatic nerves and peroneal nerves and tibial nerves in the past. Then we started doing rats. We made them smaller for peroneal nerves and tibial nerves and sciatic nerves. And then when it came time for the rat laryngeal nerve, this was too big. So we made a new cuff, smaller, custom-made little cuff, to go on the right recurrent laryngeal nerve, which lies just adjacent to the trachea, posterior to the sternocleidomastoid muscle of the rat. So the rats are anesthetized, isoflurane or ketamine, xylazine, whichever we have available at the time, I guess. And we make an incision, sternum up probably about 1.5 centimeters over the ventral portion of the neck, and then by bluntly dissecting the sternocleidomastoid muscle, we open it up and you immediately visualize the trachea. And I can't get a picture of it because it's so small, but running almost posterior to the right side of the rat, left side of us, is a very large blood vessel adjacent to a very small nerve. And a small nerve is the right recurrent laryngeal nerve. And so with careful dissection and often some cursing, they're able to dissect that little thing away from the trachea. And after we do that, we get that nerve cuff around there and tie it on there. And then we route the wires subcutaneously in a rat. We go out, we put them right back in here, and we externalize them at the base of the skull in the rat because the rats will eat anything they get their hands on, including anything that's attached to their body that they don't recognize. So we attach it to the head, to the skull, so leave it alone. And we clean off the skull, we make an incision. This is dental cement. And the wires come routed out right behind that incision. And we just take an Amphenol connector from Radio Shack or Samtech or wherever you want to buy them, solder those leads onto that head connector, and then secure it with more dental cement. And then we have the ability to plug in that nerve cuff to a stimulator. And then while the rat's anesthetized, we can control the muscle, hopefully. Now, things can sometimes obviously go bad, especially when you're working with a tiny little structure and sharp instruments. So we had to find a way to be able to accurately assess that we're applying the correct voltage to stimulate these muscles, as well as the fact that we didn't damage the nerve when we did the surgery in the first place. So we modified an endoscope. This is Dr. Andriata, another speech pathologist, and we got a veterinary endoscope, and we found that we could kind of get the rat kind of semi-Fowler's position in a rat recliner. So we made a rat recliner, and that's supposed to look like Swiss cheese because it's a rat recliner. That's why he made it, I swear. And so we can't use isoflurane because we have to go down the pharynx to larynx to look at these tissues and these muscles. So we found if we sedated them first, acepromazine, and then anesthetize them very deeply with isoflurane, it would take them 10 or 15 minutes to recover, and that was enough time to scope them. So there's a rubber band that goes right behind the incisors of the rat and holds them up in the air, and Dr. Andriata scopes them, and this here is an image of what we get on the system. And this is how we determine voltage and effective stimulation. So what you're going to see, so I'll orient you as you go down. This will be anterior to the rat. This will be the rat's posterior. And when you go down, you're going to see the vocal folds as he breathes. And the first thing we look for is that the vocal folds are actually moving while he breathes, for one, because they're not, we paralyzed one, and then he's out of the study. And then you'll hear me say something about voltage and Richie saying yay, and me telling him he's getting cocky because he's really good at it. And but you'll see when they do stimulate it that you should see that vocal fold move over at the same time, out of sequence with breathing. That means that we actually got the nerve to fire and stimulate that muscle. Oopsie.
Got it.
And so,
Cool. Got it.
and that for us was a positive test. We have the right voltage then. It's not paralyzed, it's working fine. And that rat will then go into our exercise group so we can exercise that one side of the larynx and not the other one. So we instrument a lot of rats, collected a lot of stimulators, and then we did 8 at a time. And so we lined them up every day. The exercise group, and these are, by the way, the 30-month-old Fisher Norway Brown 344 age model rats. They are twice as heavy as the 6-month-old group. These guys are up to 650 grams, 660 grams, compared to 350 for the young group. So they age well. And we hook up the stimulators to the head connectors. We've already established that the system works in all these guys. And then they're hooked up to these things twice a day, 2 hours at a time, for 500 millisecond trains every 2 seconds at 100 hertz. And again, this was something we did that hadn't been done before with laryngeal muscles. So we overstimulated these guys, mostly to see if something was going to happen, because we were afraid at the beginning of this whole thing, if we went with a really more physiologic, in humans anyway, short duration exercise protocol, and we had subtle changes in the system, we would never catch it in these tiny little muscles. We went overboard in hopes that we could do something with these guys first round. And so we went with a program that's 4 hours out of every day, 5 days a week for 2 weeks. So once the rats were finished exercising, they were sacrificed to CO2 asphyxiation, and then we took out the laryngeal group, the entire larynx, as a whole unit. And so you'll see there's your epiglottis right here, and in this little trachea, you'll see the left and right vocal folds are in there. And under those vocal folds lies the thyroarytenoid muscle, the TA. And so these guys are bathed in standard Krebs solution, bubbled with 95% oxygen, 5% carbon dioxide to keep them viable and happy, and glucose and all the good things that they need, because it takes hours and hours to dissect out one of these tiny little muscles, as you can imagine. And so we dissect them down and down and down to the point that we can find some cartilage that's left to attach some sutures to. So these guys don't have tendons. So they're attached to, in the anterior portion, your thyroid cartilage, and in the posterior portion to the arytenoid cartilage. And the arytenoid cartilage is about 1 millimeter tall and 0.5 millimeter wide, probably. So the arytenoid cartilage, because it sits on top of the cricoid cartilage and pivots, we kept some of the cricoid cartilage to suture to. So we're suturing to the cricoid cartilage and the thyroid cartilage, not any tendon and no bone. And that is one thyroarytenoid muscle. It's about 3 millimeters long, and they weigh about 3.5 milligrams. So I don't know if you all have a system like this here now. I can't remember if you did in the past. This is an Aurora system. I think there was a Cambridge system once upon a time. Cambridge became Aurora after a while. Aurora's out of Aurora, Ontario. And they put these systems together for you if you don't want to make them all by your own from scratch. So there is the chamber here that is kept at whatever temperature you want to by the water heater / water cooler. The lever system is up here with the force transducer and the stage, and the tissue sits in the middle between 2 platinum electrodes, which will be super maximally stimulated by the stimulator that Aurora gives you when you buy the system. We did this at 25 degrees Celsius. Some people go at 37 degrees Celsius, but at 25 it's been shown that the tissues last longer and are more viable. So we wanted to keep these around and functioning as long as we possibly could. That is one TA muscle attached to the thyroid and cricoid cartilages, surrounded by those 2 platinum electrodes. And I think, so we did an optimal length test first, like we always will. We'll slowly increment the length of the muscle until we get the peak force. I went with the John Faulkner method of going with peak force of twitch force, because I didn't want to fatigue these guys too much while I was trying to do this, because I've already had these guys out for a couple hours at a time. So we found optimal length. Once we did that, we gave them to a force frequency test. So every 2 hours, every 2 minutes, they will get another stimulation of 1, 15, 30, all the way up to 300 hertz, so we can produce a force frequency relationship with these tissues. And then we'll do a fatigue protocol. This is a fatigue protocol from Mike Reed that he had done for a long time that induces low frequency fatigue in skeletal muscle. We thought we'd try it in laryngeal muscle. It's a 40 hertz, 500 millisecond stimulation every 2 seconds for 150 contractions for 5 minutes. After we collected all those data, one thing that we need to do was to normalize the forces, the force frequency and the force, optimal force, etc., and the fatigue forces down to a normalization parameter like cross-sectional area. And so normally we do this with these kind of systems with a digital caliper set up against the muscle and say this is how long it is. These things are so small, I had no faith in doing that whatsoever. So what I chose to do instead was to take the muscles out after they were done and pin them to cork in the most linear position I could possibly find. So I didn't overstretch them, but I stretched them out long enough so that I'd be able to get them relatively straight to make measuring them later a lot easier. And I put them in 10% neutral buffered formalin for about a week and then analyzed them from there. The calculation for normalization for PCSA is thus. This is from Ward and Lieber's paper from 2005. And I chose Ward and Lieber's paper from 2005 because they have used a number for density to account for the dehydration of tissue after it's been fixed for an extended period of time. And so we use 1.113 grams per cubic centimeter for our density measurement. And afterwards, we then take these tissues and we will measure length, we'll measure sarcomere length, we'll find sarcomere number, and we'll see if that was influenced at all by this exercise program. So this is one of the TA muscles that we pulled out after it was fixed. Putting it linear was the best thing we could do, I think, to make our lives easier. We have a digital microscope that has a 5,000 times lens on it. And this is a 50 times objective lens to measure length, 4.2, 4.6, and 4.57 millimeters, sorry. And we get these measurements for length. And then we change the lens to the 5,000 times objective, and then we will measure sarcomere lengths as well. And the system's calibrated to automatically recognize the 5,000 times objective, so it's going to automatically measure that for you. And those measurements are 2.08, 2.08, 2.16, 1.89. These are really small fibers. Right? The cross-sectional area of these guys is about 300 square micrometers compared to about 3,000 square micrometers for skeletal muscle. It's an order of magnitude smaller in the cross-sectional area. So we take measurements at the distal, the middle, the intermediate areas, both ends of these fibers, and then we get some averages. So this right now is an N of 6, so I don't have a lot of N's. I have N of 6 right now. I don't know what the stats are on this yet. The trend right now is that the exercised TA muscle on the right side does tend to lose sarcomeres after 2 weeks of stimulation. This is the first time that I've ever stimulated the muscle and exercised the muscle without being able to either watch the muscle contract or be able to apply a load to it. And so these muscles, again, are working synchronously in many ways to produce phonation in vivo. So we're stimulating these muscles. They're pulling on the thyroid cartilage and the cricoid cartilage at the same time. They're shortening. I don't know what load they're shortening against. That is the mean of those 6. I will say that every single exercise TA muscle did lose sarcomeres. It ranged from 15 in one animal that was reduced, number 15 sarcomeres, and one lost about 360 or so. So there was a range. They did all seem to lose over 2 weeks. Cross-sectional area. Colleen did this work for us. Up here is the pure control left and the pure control right. of the muscles that the rats that were actually instrumented, but didn't get stimulated whatsoever. On the bottom, on the right-hand side, is the, I don't know why I did that. On the right-hand side is the non-stimulated, and on the left side is the right one, the stimulated side. And we do note that there is an increase in cross-sectional area in the stimulated side as opposed to the left non-stimulated side. But we do seem to have a crossover effect that's going on with these muscles because the left side as well seems to be larger than our control animals in cross-sectional area. The arrows are pointing to what appear to be centrally located nuclei. So this one up here, that one right here, and not really that central, but they're not peripheral either. And so as a guess as to whether these are centrally located nuclei, embryonic, either cells that are in distress, they're not large enough to be distressed cells, I don't think, but they could be embryonic and regenerating or relatively nascent fibrils. Sodium channel, voltage-gated sodium channel 1.5 is a voltage-gated sodium channel that is common in cardiac tissue, but not expressed in skeletal muscle unless it is embryonic or very young and growing tissue. So we use NAV 1.5 as an indicator of denervation. There's been evidence that laryngeal muscles undergo a lot of denervation and damage and recovery over the course of a lifetime. So we wanted to see about denervation. And so up here, again, left and right controls of just the control animals. And the bottom is, the left is the stimulated side and the right is the non-stimulated side. And the control tissues here, I'm sorry, the green here is actually NAV5. And you'll notice that there's a lot more of it in the control tissues than there are in the stimulated tissues, which would indicate that we have attenuated denervation in these muscles. I don't know. That's a question. Maybe we can talk later. That's a guess. I don't know. I'm not, it's not my area by any means. So this was interesting to me, but I don't know what I would interpret it as, to be honest with you. A mitochondria is a modified Grimoire stain. And you'll notice here these speckled patterns of purple. We seem to have greater incidence of mitochondria in our exercise group compared to our unexercised group. And the force frequency. So in the left-hand side, you'll have the force frequency relationship. On the x-axis is frequency of stimulation, and the y is the specific force, the normalized force to cross-sectional area. The red is the right TA muscle that's exercised. The blue is the contralateral TA. It's not exercised. And the green is the pure control animals. And I don't have stats on these yet for you because I just did them, I think, yesterday or the day before. And so, what we do know is that there appears to be an increase, whether it's significant or not, it might be, in the exercise group at high frequencies. However, the contralateral control side, with the crossover effect likely, has depressed forces at all frequencies compared. So we did a relative force and looked at it as a percentage of the peak, and the peak being 300 hertz stimulation. And we look at that, it looks as though we have shifted to the right moderate amount, the right and left TA after exercise compared to the controls, which would be a shift towards a faster phenotype. In the young rats, again, this is the very first contraction after the force frequency analysis. The very first contraction of 40 hertz. And we set everything to be percent of initial. And so you'll see some potentiation, some staircase potentiation that takes place here in the first minute or so as forces drop and then increase again, that competition between fatigue and potentiation that takes place. The green, again, is control. The blue is contralateral. And the red is exercised. And we do note that we have a reduction in endurance out here for the exercise group compared to compared to the contralateral side. There is no difference in the contralateral to control, but compared to the left unstimulated TA, the right TA is more fatigable after this exercise bout over 2 weeks compared to what it was. They also seem to be more susceptible to fatigue and exhibit less potentiation in the staircase than what the control does. And the aged rats are the ones that we're hoping to find out. I haven't got these all analyzed yet. I haven't got the control group done yet, so I apologize for that. But I didn't want to not show you what I have so far. What I have so far for the aged rats is exercised right side and contralateral left. So the red is the exercise side, again, and the blue is contralateral left. There's a boatload more variability in these aged rats than there was in the young rats. And again, we seem to have shifted this relationship upward. But when we look at the relative force on the right-hand side, there doesn't seem to be a shift in frequency of force production. Just seems to be almost overlapping themselves. So we seem to have increased the force production here in force frequency, but we don't really have a shift in that relationship at all. With fatigue, blue is, I rewrote the programs, by the way. The programs for Aurora are very, very finicky, and if you don't write them correctly, they don't give you the output. So when I had programs the first time with the young ones, I had to go in and I still have to go in and analyze every single 300 contraction for every single rat independently by hand. So that's why you only have 10 frequencies or 10 time points for that relationship. I rewrote them and now I get the whole thing. So this is 300 seconds of fatigue, 150 contractions over that course of time. Blue is left. The blue line is your standard deviation. Red is the exercised 2-week group, right side of the TA, and red line is the standard deviation. And you notice kind of a not the same trend as we saw in the young rats, but I don't have all the data for the young rats yet, and that might be an issue because we do have higher forces as we expect because we do have a shift upward of the force frequency relationship, and we do have some potentiation, but again, we don't have the same potentiation with these guys that we do with the left contralateral side. And at the end, we have less force produced, and we have an increased fatigue in our exercise group again. So right now, we have a model for exercising laryngeal muscles in a similar way as we've done in the past for skeletal muscle. The limitations of this thing are we don't get to visualize contractions. We don't get to record force production. We don't get to see kinematics of muscles. We don't get to even visualize over time how they may or may not be improving in function when they fatigue or anything. So we don't know visually and have no data on the exercise bouts themselves. But we have a way of controlling one side of your voice box, one side of the intrinsic muscles, to see if we can potentially manipulate the system and find a way to optimize these functions. So we have been able to increase isometric force almost at all frequencies. In the young ones, there seemed to be more of a high frequency shift. In the age, they seem to respond across all frequencies so far. We do seem to have reduced capacity for endurance. The phenotype of laryngeal muscles, the thyroarytenoid, like I said, TA, it's easier to say, the TA muscle is exclusively 100% fast twitch. Predominantly type 2A, which is oxidative fast twitch, but it also has 2L, and 2L was first, I think, observed about 1995. It's a whole new myosin heavy chain that is only exclusively in the laryngeal muscles and the ocular muscles, rectus muscles of your eye, because they're very fast. So the speed of contraction of a mouse TA is about 6.5 second contraction time. In a cat, it's about 12. So they're super fast. But we don't know yet what the fiber types are in these guys. We don't know yet if we've transitioned any of these. We do know the ATPA stains say they're all fast twitch. That's great, but we don't know what the isoforms are. Because the TA, although is fast, it also has oxidative capacity about it. It is 2A, mostly. The Thyroarytenoid is about 50-50, and it also has some type 1 fibers in it. So we also have a chance to look at all these muscles, but they are all pretty small. We do have a crossover effect likely. It's not surprising because we get crossover effects in limb muscles all the time. We exercise one other TA, we get a response in the left other TA. So right now we're running through all these data still. We don't have as much tissue from these guys as we do from a rat, from a cat, even from a human biopsy probably. So when we run through this many analyses, we don't have any tissue left. So we go back and we instrument more of these guys and do it again. So now we got to figure out what we're going to do next. But that's what I have for you today, and thank you for letting me talk in. This is Kentucky. We don't have mountains, but we got mist and pretty trees.
Close up from questions. Yeah, I'll start out. I don't recall if you presented whether the cross-section area of the exercise site was greater in total or not.
Right. So you're right. I didn't. It was so far. It's greater in both the young and the old. In the young, it was well over 500 square micrometers compared to about 350 in the uncontrolled. The old rats also seem larger, but we're talking about I think 400 square micrometers to about 300 micrometers on the left side. So I don't know if that's significant yet or not, but there were increases in cross-sectional area in both of those.
I think you considered that could be the cross-sectional fatigue or the blood suction.
Yeah, that the mitochondria slide I showed you is yeah, obviously when you have an image you show these are the best representative images that we have, but overall there's an increase, but it's not that large. And so yeah, we have a great cross-sectional area, so we have some more increased specific force. We've put some more contractile proteins in there, probably all super fast twitch, maybe be 2x. And so yeah, that and the size could possibly be why we don't have as much endurance as we had before.
What kind of sarcomere length range does it work in? So from a relaxed TA to a fully activated TA? What's the
so I wish I knew the question. I mean, to answer that question. The challenge is, of course, that we pull these things out, and then I'm cutting them all apart, and I slap them in the dish, and then we exercise them, and we put them on a stick, and we fix them. I don't know. There's no joint angle, right? There's a thyroid cartilage, and there's a cricoid cartilage, and I know the numbers. I know the lengths when I take them out when they're fixed, but because I don't have a reference point of what is useful, it's hard to say. It only has 1,400 on average, 1,400 sarcomeres in series, right? And of course the sarcomere length measurement is dependent on how I fix them. The group I got better at fixing and stretching them out for better fiber length measures obviously had longer sarcomeres. So I don't know what the ranges are going to be.
Do you know what the optimal length in that muscle is?
For a rat, nothing has been written. Nothing has been written on a rat laryngeal muscle actin myosin. So I'm going off of rat skeletal muscle actin myosin, and I use 2.26 micrometers as optimal. I don't know. Nobody's ever done that one yet. So if you want to, great.
I've never missed the one actually. Is the fatigue displayed in absolute or relative values?
Those are percent of peak.
So
Yeah.
absolutely, though, how did it look?
So the absolute values of the young group was higher than the old group, and I believe 40 million newtons of force was the. That'd be the. I'm getting back to the wrong equation. 40 newtons of force, I think, was what the young ones were averaging in these muscles on just a non-normalized force value.
And was the exercise quite beneficial with raising the force output?
See, that's a good question, but we don't have before and after data, right? So this is a one time out and exercise them. So
But
yeah.
relative to controls?
Yeah, relative controls, there wasn't a difference to controls. Doesn't seem to be, yeah. Because the changes are so subtle and small in these guys, but I don't remember there being a big difference. I remember initially when I was starting to analyze the young ones, I had 4 of these guys done, and I did a curve, and they were off. They were improved by about 8 million newtons of force. And then I added more data and become more variable, and then I normalized them, and that's where we got the differences here, maybe. But I don't think that the differences were that startling in that force.
Yes.
So I'm wondering what the exercise does to the laryngeal muscle function, like at the level of the sound, and especially in naps. How can you see the difference when the muscle is playing? And initially, how do you see the role of the laryngeal muscle in rats?
So in vivo, just in function in the rats? Yeah. The challenge is, of course, these muscles are covered with that vocal fold net mucosa, right? So even when we visualize them and they're doing their thing, we can't see those muscles. There's no way to see them. One of the reasons we've gone to this as well is because you can't biopsy humans' vocal muscles, because if you do that, people become immediately hoarse and they can't talk anymore. So the function part is a challenge because in vivo, I'm still trying to kick around ideas of what else we can do besides this kind of a thing, because I'd love to do something different with these guys. But if you try to put in a transducer or something and then stimulate them and have the force produced before and after even, you're including their breathing. Maybe you can do that for a couple seconds, I don't know. But we haven't been able to. We're going off of these sort of data and interpreting this as an improvement or a decrement in function. Yeah.
So if you have a laryngeal muscle which is relatively sedentary-ish,
Yeah.
you exercise it 4 hours every day, super maximum activation, do you think it's possible that you've overdone it, maybe then causing higher fatigue?
Yeah, absolutely. We did some pilot data initially with some rats, and we used the typical stimulation protocol, but we used for limb skeletal muscle in rats. I mean, it was 250 hertz, and it wasn't 2 hours though. But we got done with that, and the fibers were smaller. And people are like, "Oh, it's an endurance fiber." I go, "No, it's a really angry fiber." I think that one we really fatigued the crap out of them. And I think we drove at such a high frequency it wasn't beneficial, and then we dropped it down to 100 hertz for this one. So I think we're still not close to the optimal yet that we're looking for. I say that because when I look at these, I still see evidence that there are areas where it looks like there are fibers that are missing. Possibly there's greater endomysial space that may have been inflammation once upon a time in the last 14 days. So I think that there's a lot of remodeling going on that might be better suited to be repair and adaptation down the road. So I think we're still going to drop it down a lot more.
Do you know if these left and right laryngeal muscles during normal function, do they usually work in complete synchrony, or do they work independently of one another?
I asked that question to our speech people too. Yeah, that's a good question. Allegedly, they work in synchrony. Now they're driven by 2 different nerves, right? The right recurrent laryngeal nerve does the right side, but it innervates all except for the cricothyroid, which comes off a lower branch of the vagus nerve. But they all come from the vagus nerve originally. The left side we didn't use because there's too much crossover to other muscles, and nobody, I think, ever told me what those other muscles might be. So even though they're supposed to work in synchrony, the nerves that drive both sides are identical. And so there could be a lot of differences that take place, but I don't think anybody knows that yet.
Yeah. With this exercise protocol, do you know if you get any cartilage remodeling? Do you see any changes in the anchor points?
Oh, that's a good question. I haven't seen any, but I haven't looked for any either. Because to be honest with you, when I first started doing this looked like exactly what it looked like to you last night, which was a rose in a petri dish, right? And trying to work your way around this thing, cutting off all this extraneous tissue. I'm just happy that I get to the end of it, that it's still intact a little bit. So I never really even looked at it yet. But if this thing is contracting the way it looks like it's contracting in that scope, and it does that repeatedly for that many hours, I would assume there's remodeling of that cartilage. And I don't know what that would mean for function. Yeah.
Brandon in the middle.
Do you ever think about doing an animal that actually make sound like Canada and meows, let's see if that has resulting changes in the actual,
Yeah.
I don't know, vocal patterns or vocal cords or sounds that we're getting.
Yeah, that's a good question. And is a group. The other group that does a lot of laryngeal muscle biology is University of Wisconsin. They have a lot more lakes than we have, and it's cooler and it snows there. So I might want to go there one day and visit. And I forgot the guy's name. I'll find it if you want to know. He came down and gave a talk, and rats actually do vocalize all the time, right? You just don't hear them. The DLAR, for instance, in Kentucky, if you want to go in there with a set of headphones, they'll let you go in there at night in the headphones, put headphones on, stand there, and you'll hear these guys talking all night long, really loud, right? And during mating season, they go insane. And so his idea was he trained, how do you do this? He trained a male rat during mating season to keep vocalizing by giving him treats every time he was annoying the girl next to him. And by giving him treats every time, he got this rat to constantly make these sounds you can hardly ever hear. And then had sort of a vocal training response with this guy. So he and I chatted briefly about being able to do something like that and then study the muscles more physiologically again, because then at least these things are talking more than normal. But I did see a presentation on sheep when I was at Wisconsin last, out of Austria. They're doing some sheep work. I think because the muscle's bigger, not because they talk more, though.
I actually wanted to follow up on things that Tim mentioned, because I was thinking, at the beginning you started out with this problem that somehow these muscles don't close that gap that they're supposed to close. And the idea is that there might be muscle weakness, and it happens more in the elderly compared to young. And I was wondering, could it be that actually has nothing to do with the muscle, but it has to do with this cartilage frame that's hanging there, so that's actually has remodels in age, either that its structure is different or that its mechanical properties are different, and despite the fact that we still exert the same amount of force, now it just has deformed somehow, or it's stiffer, and so it's got nothing to do with the muscle, but it might have to do with the cartilage where it hangs.
Yeah, that could be. It's a very good point, and it might, because we do know that cartilage properties change as they get older, right? So I don't know if anybody's looked at that. I mean, so little has been done in the basic science world of these muscles and laryngeal muscles, et cetera, and they're articulating the cartilages that articulate when you speak. But I will look for that. It's a good point. It would make sense if that was the case too. Again, all these observations are coming from the clinic, right? There's no biopsies, there's no tissues to look at in humans. So I don't know.
The other thing I was thinking about force measurements in these muscles, and you're mentioning you're introducing a little. Well, I was thinking you could introduce a little balloon and then measure how much is pressurized. Then you were saying, well, it's a problem you can breathe. And I was just wondering, it should be possible you just open up the airway below.
Yeah.
And so that you actually would breathe through, here.
Right.
And then the laryngeal muscles are higher up, and so you could introduce.
So just include that and just measure that. Yeah, and that's a possibility.
I mean, not in humans, I would suggest.
Human, but then it's not translatable. No. Okay. No, that's true. I
mean, if you really want to get at a force in the muscle.
Yeah.
The other thing I was actually interested in is that so these muscles, when they lay there and then they get together like that, when they contract and they seem to get together, I assume the fiber direction is along the long axis of the muscle.
For the TA, it is, yes.
Okay, and so really what happens then is that these fibers are relatively slack, and as they're taken up, then it becomes stiffer, and then it becomes more a straight line.
Yes.
And then they get together. That's how.
Yeah. So the natural aperture is to be open, and that's to pass the tension. And then when these muscles contract, they will pull them in and tension them. And tension them is what gives them the resonance and the frequency that you're looking for. And the TA and the cricothyroid are the tensioners of that muscle. So they don't close it, but they work to make it tighter or looser depending on what frequency you want.
And so I was also wondering, you determine optimal lengths with a single twitch? Or
Yeah, the twitch. Yeah. the thing I used to complain about all the time back in the old days. I did it.
So that probably would mean that your peak is actually shifted probably to
Yeah.
longer lengths.
Absolutely.
Because of some maximal activation. And
Yeah.
I was wondering when you then do the training and you lose sarcomeres,
Mhm.
whether or not that will have an effect, and if you actually might be working at two different sarcomere lengths when you do the mechanical testing, or in the mechanical testing, do you independently verify the peak force with a high frequency?
Mhm.
You know what I'm saying?
Yep. So we do a 300 hertz stimulation in between every single frequency, interspersed between the two, as a test of muscle rundown and the fitness of the muscle. So we do a 1 hertz, 2 minutes, 300 hertz, 2 minutes, 15 hertz, 2 minutes, 300 hertz again. We go back and forth with that. And at the end of the time, we have these 10 300 hertz contractions that we then look at and make sure that that's still linear. And if it deviates at all, then either there's compliance in the system, it's shifted, or we have fatigue or damage, or something has gone wrong.
So you mentioned that the 30-month-old rats are a lot bigger than the young ones.
Yeah.
Is that mostly fat?
Yeah, almost exclusively fat, and fat infiltrated in the muscle as well.
So do you see more fat infiltration in those muscles than the older ones?
We in the limb muscles we do. Yeah.
You don't in the muscles?
We didn't in the skeletal in the laryngeal muscles. No, which is interesting. That's a good. I never even thought about that until you just mentioned it. We don't see any fat in there at all. But yeah, when we've done these guys with skeletal, we do see that. Yeah.
Do they always fail to a particular side? Or does the failure occur sometimes to the left and sometimes to the right, or is it always to the right?
Oh, and just in general?
Yeah.
I don't know. I think I never asked that question to our clinical folks, and they never mentioned it. Not sure. I'll make an assumption that it would be. That's not even a good assumption. I have no idea. Let's go leave it at that.
So you mentioned the clinical scenario. Is the incidence of some of these dysfunctions the same in males and females?
Presbyphonia and presbylarynx is greater in males, but I don't know the numbers. I remember that conversation with Joe in those presentations, but I don't remember the numbers of them. But it is greater. In Kentucky, we have a great thing called tobacco that was grown there forever, and we have an amazing number of cancers of the neck, and that too is more prevalent in males, and that too is something that we're also trying to induce in rats now to see if we can do something exercise with that.
Do you still get the brown warheads from NIH?
Yeah.
Because we don't get them anymore.
No?
No, because we were on a protocol with NIH and the whole world got it from them.
Yep.
But, hey, was it 2 years ago? 2 years ago. 2 years ago, they don't give it to foreign countries anymore. So we potentially would be very interested.
I have it in.
Issue.
Yeah, no, it's good. Yeah,
that
absolutely.
you might not use.
We aren't using, as you can see, a lot
Not
of it.
if you're allowed to smuggle it across the border. Come after us.
The rules say that once the animal is disposed of, that tissue is tissue to go anywhere.
Right.
Yeah, so we're happy to
Because
send it.
you're actually, because you get them for free when you have the protocol.
Yeah, we get them all free.
And we actually were willing to pay for them.
$600 a piece.
And they don't do it. Yeah, at the moment, in Canada or anywhere else, you can get those.
Yeah, they don't give them away, like you said, right?
We
So,
started some interesting work with Jeff Lauer. And
yeah.
couldn't finish it up.
Oh yeah. So, well, our grant is over. We've done to 162 of these rats. And all the protocols that we've done amongst the pilot data and other things that we've done. We also done growth factor injections too in some other studies as well. So we will get more. I mean, we're writing more grants, and when we get an NIA grant and we get back to these rats again, Esther and I also have studies on rats as well, NIA rats and the same rats for different studies. So yeah, we will. If you want rats, we'll send you all the pieces of parts we have.
Thank you.
Yeah, sure.
So the other thing is, your specific forces there for the younger 6
Yeah.
newtons per square centimeter and 3.5 for the old.
They're small.
Yeah, they're very, very small.
Yeah.
Do you have any explanation why they are so low?
I don't, and so what happened was I got done and I had a presentation in Wisconsin, and I had values of 25 and 30 newtons per square centimeter on these guys, and one of the laryngeal biologists said, the stress of a dog TA muscle is less than what you're presenting right here today. How do you explain that? And I go, I can't, and but I'll try to figure it out. And so I went back through Aurora's system again, and I went through all the data again, because it drives me nuts when things give you numbers, you don't know where the numbers come from. And I noticed that in Aurora's data acquisition system, when you're actively calibrating the system and running the system, it gives you millinewtons of force. Then when you hit the button that says analyze, it gives you grams of force. And I'm like, that's an order of magnitude difference right here in this whole thing. But the numbers didn't appear to be different. If it was 10 millinewtons here, it was 10 grams here. And that's what happened in Wisconsin. I got a hold of the Aurora folks, we went through the whole thing, and sure enough, that was a software problem they had for 20 years. Nobody ever bothered telling them. And so we were an order of magnitude too high. And when I went back and did all the data again, I'm too low. And so that's what the numbers are. And I don't know what the biology of these muscles would be, that they would be so much lower than most muscles would be, especially skeletal muscles.
Yeah, we have a similar instance, where we find very low forces in some muscles, well, myofibers and MDM, fibers from MDM.
Yeah.
And so our first thing was that we are not really sure about these very small diameters. And so how do you actually calculate cross-sectional area for the normalization? How do you get that?
Yep, that is the mass of the muscle, and that is then we have the density and the length of the fibers, and that length is, you can see, best I can do is magnify it by 50 times and try to make it as straight as I can.
So that's how you do it. You don't measure a.
No, because I tried that initially. I used the digital calipers that were sitting right next to the unit, and I sit there and I'd go like this, and I'd say maybe it goes up a little more. I go up by a little bit more, and it changed it by 50%. And I'm like, well, it's just not really that reliable. So
the other thing that I thought was really interesting. So these muscles have absolutely no dystrophin.
they have very small amounts of dystrophin, and so if you stain for it, you'll get a faint little red line, but you won't get the big red line that outlines every single fiber like you get in skeletal muscle. But if you do utrophin, you get that dark red line that outlines every single fiber. That's going to anchor it, but very little amount of dystrophin, mostly all utrophin.
And do we know why that is? Or are there other muscles that?
The rectus muscles of the eye as well. All the cranial muscles, it seems, I can't say all the cranial muscles because I don't know that. Of the muscles that I know that hold utrophin are muscles that come from cranial. And so those are rectus muscles of the eye, and right now the laryngeal muscles. And I don't know if other muscles from the cranial origin are the same way or not. They also have different expressing satellite cells. Satellite cells from these muscles have been tried to be implanted into skeletal muscle, I believe, once upon a time, and doesn't work as well because they're different satellite cells.
And so utrophin is just a transmembrane protein, like dystrophin that connects.
Exactly. But it's subtly different, but it's different enough that it's not affected by the loss of dystrophin protein in MDM. Yeah. I learned that about a year ago. I was fascinated by that. That and the 3 innervations on that one muscle fiber were the 2 things that I was like, wow.
And so a regular skeletal muscle then would have corresponding less utrophin?
Yes. I don't think there's any utrophin in skeletal muscle. That's what I found. I haven't found a paper yet that talked about utrophin in skeletal muscle.
And then the other thing that I thought was really interesting was, the 2 or 3 neuromuscular junctions.
Yeah.
So why do you think that is?
I talked to Dr. Andriotta about this, and I said, can we trace these back and see where they go? Because I want to know where these alpha motor neurons, if they're alpha motor neurons, which they're supposed to be, are coming from. If they're coming in this, are they members of the same motor unit? Are they different motor units? Is it a depolarization event that occurs across the entire membrane that would spread the action potential faster across the whole membrane? Or is it an evolutionary aspect where these tissues are so important to survival of a species that if you lost one nerve, which happens in aging, you can still contract these muscles? because you have multiple neuromuscular junctions. And those are my two thoughts. I don't even know if they're salient thoughts, but we hope to be able to trace these things back and see where they come from. See if that would tell us.
Could it have potentially anything to do if you have to activate, deactivate the muscle fairly quickly? You want to change, you know, the resonance of it, the stiffness of it. That
could be.
You have multiple neuromuscular junction that you might be able to do that.
Maybe, and then I don't. Would they come from the same motor unit? Do you think they'd be branches of the same motor neuron, or do you think they would then have to be different motor units and drive it?
I mean, I don't know. And you don't know that. You don't know
Right.
if at the neuromuscular junction you might actually innervate the same motor unit.
Right. I have no idea. Nobody knows. I had that conversation again with the folks in Wisconsin, and still nobody knows. So
That should be, I mean,
Doable.
not easy, but relatively easy to
Yeah.
actually determine.
yeah, I agree that based on, I even talked to Andrew about it this morning about it. We should be able to track those things back pretty quickly. That technology is there. It's just everybody in my college is clinical. Their research time is minimal, and trying to get the research folks on board that have so much clinical and teaching time to say, just let them play these rats for a while. Sometimes takes a while.
Tim and I wanted to do some work at one point on cat tendinosis, and it has two neuromuscular junctions, and then it has this funny connective tissue band in the middle of the muscle. So it's considered one muscle. Everybody calls it the semitendinosus. But it has a muscle belly here, and I don't know, 1-2 millimeter connective tissue sheet, and then another muscle belly here, and then you have a neuromuscular junction there and one there. So it looks like two separate bellies just divided by a little bit. But your muscle is not divided by little connective tissue sheets of longitude.
It's not. It is exclusively, and this is Feng's group, and if nothing else, they have amazing pictures. It's a great paper, but yeah, this right here. So that's one fiber, and it's multinucleated, and the nuclei are where they're supposed to be, and then you have the neuromuscular junctions. There's 3 of them right there in that one. So.
And so they are really, I assume they're just like a millimeter or 2 apart.
Yeah, not far at all. And this was a mouse, and so the fibers of the rat, 3 or 4, depending on how you fix them, 3 or 4, I'd say 5 millimeters at the max to stretch them out really far. So that might not even be a millimeter in this mouse.
Great. Going back to the idea of measuring the function in vivo.
Yes.
You said the problems of measuring the force produced was that you have to occlude the larynx and it would stop the rapid breathing, right?
It would be if you wanted to put something in there to squeeze it, yes.
Yeah, but I know that in mechanical respirators, when people are in ICU, they have like a cuff for the tube that they insert into the trachea.
Okay.
And so I was wondering if would be able to produce some
I see what you're saying.
mechanism like that,
Yeah.
because it's like a cuff with a tube in it. So the airway is still unoccluded, but you can still measure the pressure that is being applied to the cuff.
Okay.
And it's actually something that they do measure like really grossly with a tube that comes out of it, so you can measure the pressure of that cuff.
Okay.
So maybe that would be a possibility.
Yeah, no, that's I like that idea. That could be fun to do. Thanks.
I noticed that the laryngeal muscle was closing during breathing. Do you think that the heavy breathing of normal exercise training could have a training response on the larynx?
Let's see. I'm not sure. I really don't know. I mean, it would obviously increase the frequency of contraction over time. I mean, if it was going to have anything, I think it would have something to do with an endurance effect, possibly. But I'm not sure. Again, I know nobody's ever looked at that, but I would assume if there's going to be something, it would be a possibility of improving its endurance capacity, because it's going to contract for repeated times more quickly, right? So that could be.
So I'm curious, in a clinical setting, when you see patients with a situation where the larynx isn't quite closing up, what do they actually prescribe in terms of like exercise or rehab in that situation?
So what you heard in that video earlier with Joe, hitting the tone and the guy trying to match that is what they do. And they send them home and they have them doing voice exercises. And it's not all day long, but I think it's about 20 minutes at a time. And so that they can rest and everything else. Because it is apparently pretty taxing on these people when they're trying that hard to hit a note. But it's just a matter of pitches and also loudness. And to give me, there's 3 or 4 things. There's pitch and there's volume, and then there's 2 other things that they try to do. And depending on where in the system that the vocalization is broken down, because it's made up of respiration, resonance, and the other one, respiration. And so, I might have said that already. So it depends on what, it's different exercises for different aspects of the 3 portions that make up speech. With that, it's going to be the resonance issue, so I think it's more pitch than anything else to try to get. But it's not 2 hours at a time twice a day.
Any final questions for Dr. Butterfield? Thank you very much for your presentation.
Thank you. Thank you.